Hello, Dr. Mirko Pegoraro, and this is your seventh lesson of the Drosophila of the developmental biology module. So today we are going to talk about the tetrapod limb development. The tetrapod limb development is common, the development of the limb is common to all vertebrates, from fish to birds to reptiles to mammals. So they all have the same structure. Why they are called tetrapod? They are called tetrapod because they have 4 limbs. In terms of what we are going to do today we are going to talk about the formation of the limb bud. We are going to talk about the axis of the limb, so how the different axes are formed in the limb. We are going to talk about, of course, about the different genes that are expressed and forming those axes and how they interact with each other. In the last lesson, if you remember, we talked about the derivatives of the intermediate mesoderm and the lateral plate mesoderm, as well as the derivatives of the endoderm. In the next lesson, we will going to talk about metamorphosis. Now, in the last lesson, we talked about the development of the kidney, which is a derivative of the intermediate mesoderm. We talked about the formation of the heart and the circulatory system, which are derivatives of the lateral plate mesoderm. And we talked about the formation of different internal organs, including the lungs, the pancreas, and the liver, which of course are derivatives of the endoderm. Okay, so in today's lesson we're going to talk about the tetrapod limb. So the first thing to do is to define a bit of anatomy of the tetrapod limb. First of all, we have an extra axis that we need to discuss when we talk about the limb, and the axis that we need to discuss is the proximal to distal axis. So in all tetrapods, we have the same bone structures, and the bone structures and the bones in these bone structures are divided in 3 elements. The stellopod, and you can see the stellopod here in blue, and it is the same for a primate, maybe a human, birds, and this is a horse. And you see that the stellopod is near the body, the main body of the animal. So it is proximal. So the stellopod is the part of the limb that is near the main body of the animal. This part of the limb contains usually the humerus or femur in terms of bones. Then in the middle of the limb we find the zygopod. The zygopod contains ulna and radius or tibula. Tibula depends whether we are talking about the forelimb, so the upper limb, or the front limb, or the hind limb, which are the back limb or the posterior limbs. And then far away from the body, so distally from the body, we have the autopod. And the autopod contains the bones of the carpals or tarsal, depends again if you're talking about the forelimb or the hind limb, and we have the digit. So this extra axis that we never talked about before is the proximal distal axis, so from near the body to far away from the body, and the limb can be divided in the stellopod, which is the part near the body, the zygopod, which is in the middle, and the autopod, which is far away from the body. Now, another axis that we need to define is the anterior-posterior axis. Now, the anterior-posterior axis is kind of easy to be defined. If you extend your arm laterally and you look at your hand, you will see that the first digit, which is the thumb, will point over your head. So that region of the arm or the limb is the anterior part of the limb. Opposite to that, we have the fifth digit or the pinky, which is pointing to your feet. That part of the limb, below there, is the posterior part of the limb. So now we have defined proximal to distal, so proximal to distal axis and anterior to posterior axis. In this sketch, we have a nice cartoon of a chicken embryo. In this chicken embryo, we recognize the developing brain that elongates in the neural tube along all body. We have this rectangular shape, which are the somites, and we talked about the formation of somites a couple of lessons ago. We recognize the near the head, we have the cardiogenic mesoderm, so it's the mesoderm that is producing the heart. Above that, nearer to the head, we have the pharyngeal mesoderm, which is that mesoderm that is going to, that is around the region where the lungs are formed. Below the heart, we have some budding, and this budding represents bilaterally at the two sides of the body the four limb fields, so the region of the body where we have the formation of the limbs, of the four limbs, and below that, almost where the somite finished to be produced, we have budding, other budding, which represent the position of formation of the hind limb. So now, in this second cartoon, we can understand why the thumb or the first digit is actually anterior, and in the posterior is represented by the pinky. If you look on this cartoon here, which represents the budding out of the limb, you see that this budding out of the limb has an anterior region and a posterior region along the anterior-posterior main axis of the body. And this anterior region is the region where the thumb will be produced, while the posterior region is the region where the pinky will be produced. And we will see that later on in the lesson. We also have a dorsal to ventral axis, which it is the axis that we didn't talk so far about. And again, it is quite easy to identify that axis. If you extend your hand from your body, you see that the knuckles point over your back. So therefore, that's the dorsal part of the limb, while the palm points over your belly, and that's represent the ventral part of the limb. Now, in the developing limb bud, there are three important regions. We have the progress zone, which it represents the majority, the main region of the limb. And it contains mostly mesenchymal cells, mesodermal cells, and it undergoes a lot of cell division. We have a posterior region of this progress zone that is called the ZPA, or the zone of polarizing activity. We will see how this ZPA is important, in particular for establishing an anterior-posterior axis. And we have a thickening of the external ectoderm, which is called the apical ectodermal ridge. This apical ectodermal ridge is extremely important to establish both an anterior-posterior axis as well as a proximal to distal axis, and it also contributes to dorsoventral division in the limb. Okay, but where the cells that contribute to the formation of the limb come from? Well, the cells that contribute to the formation of the limb come from different places. They come from the lateral plate mesoderm. So here in this cartoon, you have a section of a virtual section of embryo. So we have dorsal, from dorsal to ventral. We recognize the neural tube or spinal cord, as they call it here, the notochord below it. We have this triangular structure, which is the somite. In the somite, we recognize, of course, the myotome. And if you remember from a couple of lessons ago, when we talk about the somite formation, the myotome is the part of the somite that form muscles. Below the somite, we have the pronephrons. Therefore, if we have pronephrons, which are part of the kidney formation, we talked about kidney formation last lesson. If we have pronephrons here, we have this embryo is quite early during the development because pronephrons are formed quite early during development. We recognize also the lateral plate mesoderm, and the lateral plate mesoderm is recognizable because it is divided in two regions with the column in the middle. So some cells come from the lateral plate mesoderm, so migrate from the lateral plate mesoderm. Some cells migrate from the myotome, and of course the cells that migrate from the myotome are going to form muscles. Some cells come from the ectoderm, the surface ectoderm. Now, the cells that come from the lateral plate mesoderm are going to form the bones of the developing limb. If you remember, we mentioned this in last lesson, saying that the lateral plate mesoderm is responsible for the formation of different things, including the cells including the bones of the pelvic region and the limbs. Again, the myotome provides cells that form muscles, and of course the external ectoderm, here in blue, provides cells for the epidermis of the growing limb. Now, it should be not surprising to you to know that the main direction of cell division, so proliferation of cells in the limb, is the proximal to distal. After all, the limb needs to grow from the middle part, so from the trunk, outside. So the main growing direction will be proximal to distal, so from the trunk to outside. And we already mentioned this, but the limb part is organized in three very important domains. So the progressome is the main body of the limb. It is where a lot of cell division is happening. So we have cells that proliferate at high rate. Most of the cells there are mesodermal cells, mesenchymal cells as well. I mean, they are mesodermal mesenchymal cells. A posterior region of the progressome is the zone of polarizing activity, or ZPA, and this is important for the determination of the anterior-posterior axis, so it gives structure to the cell in terms of the anterior-posterior axis. And then externally, there is the ectodermal, the apical ectodermal ridge, which is a thickening of the external ectoderm above the region where the limb is growing. This is important for maintaining proliferation of the mesenchyma below it, but also to interact with the ZPA to establish the anterior-posterior axis and to divide the limb in a dorsal and ventral region. How the limb is actually induced during very early development. The induction of the mechanism that induce the limb formation in early development is composed of 4 stages. So the first stage of this is the mesoderm need to be made permissive to produce a limb. And there is a difference between the forelimb and the hind limbs. Now, what are the signals that are important to determining whether the mesoderm is permissive or not. Well, these signals are retinoic acid and FGF8. In fact, it is the interaction between retinoic acid and FGF8 that establish the region where the forelimb can be produced. Now, first of all, it should not be a surprise to you by now that retinoic acid and FGF signals, FGF8 in this case, are interacting to each other. We saw this many times already. When we talk about the anterior-posterior axis formation, when we talk about somatogenesis. So retinoic acid and FGF8 usually create two opposite gradients. We have an anterior to posterior gradient of retinoic acid, and we have a posterior to anterior gradient in the embryo of FGF8. FGF8, however, it is indeed expressed caudally, so posteriorly, form a posterior to anterior gradient. But if you remember, during last lesson, we said that BMP signaling were inducing the lateral plate mesoderm to express FGF8 in the cardiogenic mesoderm. So the cardiogenic mesoderm, so the mesoderm that is produced in the heart, also express FGF8. FGF8 inhibit the formation of the forelimb. The forelimb indeed require retinoic acid expression. Retinoic acid is actually expressed in the anterior somatic and presomatic mesoderm. So, because of the interaction between the retinoic acid and FGF8, which now is expressed not only posteriorly, but also from the heart, this interaction between the two define a region where, actually a quite small region, where there is enough retinoic acid for to permit the development of the forelimb from the mesoderm. So the FGF8, as I said, inhibit the forelimb formation. So only a small region now of the lateral plate mesoderm receive enough retinoic acid so that it can become permissive to produce the forelimb bulb. Now, the most attentive of you will realize immediately that this mechanism to define the position of expression of the forelimb cannot be the same for the hindlimb. And this is because in the posterior part of the body, we have higher expression of FGF8, and therefore we have almost no expression of retinoic acid. In fact, it is not known what is the very early determinant of the position of the hindlimb in invertebrate, or what permits the lateral plate mesoderm in the posterior region of the body to become limb. What we know is that retinoic acid is not necessary in that region. The second stage, or the second step in terms of forelimb and hindlimb definition, regard those genes that are expressed almost immediately after the initial definition of the region that are permitted to become forelimb or hindlimb. There are two particular genes that are expressed in the forelimb and the hindlimb that are the very early sign of forelimb and hindlimb determination. For the forelimb, retinoic acid induced expression of TBX5. And TBX5 is one of the very early determinants of forelimb formation or identity. In the posterior limb, however, instead of TBX5, we have expression of TBX4. And in fact, TBX4 is expressed because of islet1. So islet1 controls the expression of TBX4. And in fact, it is islet1, it is one of the very early signs, if you want, early determinant of the hindlimb formation. So the cells that will become hindlimb will express islet1. Anyway, no matter whether the cells are expressing TBX5 or 4, both TBX5 or 4 induce the expression of FGF10. So FGF10 is extremely important because it stimulates the mesodermal mesenchymal cells of the hind and forelimb to start proliferating and dividing and proliferating. So FGF10 is a signal for proliferation and therefore stimulate outgrowing. Another important step, which is connected in fact with the fact that these cells now need to start to proliferate, is the epithelia to mesenchymal transition, which is driven by TBX5 or 4, depends on which thing we're talking about. Now, the cells that are supposedly now start to divide a lot are part of the somatopleura. The somatopleura, we talked about the somatopleura in the last lesson. The cells of the somatopleura, they are epithelial cells. What does it mean? It means that these cells are binding together very tightly. So even if they divide a lot, stimulated by FGF10, they don't go anywhere because they bind to each other a lot. So what needs to happen if you want to have outgrow is that this cell need to be relaxed. So at this point, stimulated by TBX5 or 4, the cells transition from being epithelia to be mesenchymal. Mesenchymal cells are still interacting with each other, but in a more relaxed way. So when they divide, they can divide, they proliferate, and not stay attached to each other, but they are more flexible spatially. So now outgrowing becomes possible. Most importantly, and actually not most importantly, but very importantly, we also need to form, the cells need to form two important feedback loops to sustain the cell division and outgrowing. The first feedback loop is established between TBX5 and FGF10 in the mesenchyma, the mesodermal cells of the progressome. TBX5 induces the expression of Wnt signaling, Wnt2 signaling, and this Wnt2 signaling induces the expression of FGF10. And we saw that in the slide before. But what we didn't really realize in the slide before is that FGF10 is able to positively control the expression of both Wnt2 and TBX5. So when FGF10 is activated, it increases the expression of TBX5 and Wnt2, which on the other hand increases the expression of FGF10. So now we have a positive reinforcing feedback loop between these three genes that sustain each other's expression, and in doing so sustain proliferation of the cells of the mesenchyma in the mesoderm, just below the ectoderm, and so sustain outgrowing. This is what is called a positive feedback loop. And a second extremely important feedback loop is also established pretty soon. And this is, if anything, is even more important than the one we just mentioned, because it connects now two regions of the growing limb, which is the mesenchyma, mesodermal cells, with the ectodermal external cells. In fact, the FGF10 that is produced by the mesoderm, by the mesenchyma mesodermal cells, induce the external ectodermal cell to produce the apical ectodermal ridge. And the apical ectodermal ridge is an important signaling center. So what's happened there is that the mesodermal FGF10 induced the ectodermal expression of Wnt3. So the mesodermal cell express FGF10, which is inducing Wnt3 expression in the ectodermal cells. Now, Wnt3 in the ectodermal cells, we also induce the expression of FGF8 in the ectodermal cells. And these two signals make the cell change in shape. They become more elongated. The region become thicker, and this defines anatomically the apical ectodermal ridge. One thing that is important to remember at this point is that the FGF8 produced by the apical ectodermal ridge cell filter, if you want, diffuse to the mesodermal cell below them, and sustain, if you want, the expression of FGF10. So there is another positive feedback loop between FGF10 and FGF8 now, which connect the mesoderm and the ectoderm. In this way, this second feedback loop sustains cell division, mitosis, in the mesenchyma, mesodermal cell below the apical ectodermal ridge, and promote limb outgrowing. Now, why this apical ectodermal ridge is so important? Well, the apical ectodermal ridge is a signal center that provides a positional structure to the cells, so they are able to differentiate in different parts of the limb. First of all, as we saw just now, it is important to maintain the proximal to distal axis and outgrowing. It is the apical ectodermal ridge is also important because of its interaction with the ZPA, so the zonal polarizing activity. We will see that in a minute. And this will establish an anterior to posterior axis. Finally, the apical ectodermal ridge not only specifies the anterior to posterior axis, but also is provide information to specify the dorsal to ventral axis. So the apical ectodermal ridge is involved in establishing and providing information for all the three main axes of the